How do rattlesnakes see in infrared, and what purpose does it serve? Certain snakes are enabled to perceive infrared light by producing a protein called TRPA1, as well as a specific organ, the pit. This protein plays a key role in certain snakes' hunting capabilities, particularly at night, and therefore sustaining themselves. This serves as an example of why certain snakes have evolved in such ways as to why their genetic makeup has been changed to allow for the production of TRAP1. The process of producing TRPA1 all begins with DNA. So let's zoom in. DNA consists of two helical strands that are each composed of large numbers of nucleotides, the monomers of DNA. Each nucleotide has three components: a nitrogen base, a sugar dioxide ribose, and a phosphate group. Portions of the sequence of the nucleotide base pairs in the DNA can be referred to as genes. The instruction or code responsible for the types of protein produced in any given organism. In this case, the hypothetical gene that is being observed is TACGAC CGT CCA ATG ATC.
While a DNA molecule contains all of the information necessary for forming any protein which its contents enable, this process could only be performed once on the original molecule, and therefore only a very limited number of proteins could be produced. Transcription: the process in which a temporary copy of a portion of the sequence of nucleotide bases in DNA is formed. This allows the original DNA molecule to remain intact and therefore form a comparatively larger number of proteins to be produced. 20 different proteins, called transcription factors or RNA polymerase, work to separate the two strands of a DNA molecule from one another. The RNA polymerase then uses one of these strands to form a new strand of mRNA, which will have a sequence of nucleotide bases complementary to that of a portion of the DNA. However, the base pairing rules for RNA nucleotides differ slightly from that of DNA nucleotides, with thymine being replaced by uracil. As a result, the hypothetical mRNA strand corresponding to the gene TAC, GAC, CGT, CCA, ATG, ATC would be AUG, CUG, GCA, GGU, UAC, UAG. Once the mRNA strand is formed, it detaches from the strand of the DNA molecule, which then joins with its original complementary strand, leaving the DNA molecule in a state nearly identical to that before transcription. After transcription is complete, the mRNA strand leaves the nucleus, the location of transcription in eukaryotic cells, and enters the cytoplasm, or more specifically the ribosome, the location in which translation occurs.
Once the mRNA strand arrives at the ribosome, it is bound to the organelle's small subunit, in the first site, also called the A site. All strands of mRNA consist of codons, sequences of 3 nucleotide bases that encode for specific amino acids. As, during the first step of translation, only the first codon in the sequence of many codons attracts a complementary tRNA anticodon, UAC, which is attached to an amino acid. The tRNA molecule that the first codon attracts is bound to the large subunit of the ribosome and joined with the codon. After the first codon binds to a complementary anticodon, they each shift one side towards the E site. The next codon in the mRNA strand, CUG, enters the A site and attracts an anticodon of the sequence GAC, whose tRNA molecule contains an amino acid. Before both of these pairs shift one site towards the E site, and before the pair in the P site subsequently exits the ribosome forms a peptide bond between the amino acid of the tRNA molecules UAC and GAC. As a result, the tRNA molecule that was previously in the A site now has 2 amino acids attached to it. This process will continue until all of the codons, AUG through UAG, in the mRNA strand have been translated into a sequence of amino acids, held together by peptide bonds. The chain of amino acids that result from this translation, methionine, leucine, alanine, glycine, tyrosine, eventually reaches the endoplasmic reticulum, where it is folded into a protein. While the hypothetical DNA sequence covered in this project doesn't encode for TRPA1, a protein that consists of a very long sequence of bases, the same process which was applied to find the amino acid sequence encoded by the hypothetical gene could be applied to any other gene in any other organism. Meaning that, with the gene of a pip-bearing snake that holds the information for the production of TRPA1, the amino acid chain that makes up the protein could be determined by following the same process.